Chromatography. It's an important separation technique that many scientists use to separate different molecules from each other. There are two parts to chromatography, a mobile phase and a stationary phase.
In chromatography, the analyte is dissolved into the mobile phase, and as the mobile phase moves through the stationary phase, the separation of the analyte occurs.
For this little experiment, we're going to take the dye off of Skittles and separate it out into its different components. For this experiment, you'll need aluminum foil, a plastic bottle, a cup, a big cup full of water, some salt, filter paper, Skittles, scissors, a pencil, and either some Q-tips or some paper clips or safety pins. So what we've done here is I've taken the aluminum foil, ripped off a sheet, and then folded it up into a little square and opened up my Skittles bag, and I'm going to start to sort them into the different colors so we can then start to remove the dye. So take several of the same color and put them on the interior of this little wall structure that I've made. Just fold up the sides with the aluminum paper. Now I'm going to take a little bit of the water here and carefully, or get another glass, use a small glass and then take a little bit of water and pour that right where those Skittles are. So while the dye is coming off the Skittles, take your filter paper and your scissors and cut out the bottom of the filter paper. Cut off the bottom of it so you have a flat edge and make a rectangle out of it. You can leave the top a little curved. So you should end up something like this. About a 1/4 inch from this bottom, make a pencil line across the filter paper, like so. Then put a couple of dots on the filter paper, evenly spaced about a 1/4 inch apart. Next, we're going to need to spot our paper with the water and dye mixture that we have on our tin foil. I've decided to use a Q-tip. So I cut off one end of the Q-tip, or removed the cotton. Take your Q-tip, dip it in the orange solution, or whatever color you're doing it with, and come over to your filter paper, and then those dots that you made on the filter paper, lightly tap it several times, then let it expand, but then let it dry.
We next need to make a 1% salt solution. To do this, mix 1 cubic centimeter of salt into 1 liter of water.
Take your 1% salt solution and put about 1/8 of an inch in the glass, covering the bottom. Fold the paper in half so you have a little bit of a crease. This will help it stand freely. Then put it into your developing chamber with your developing solution.
After a while, you'll slowly see different colors emerge. As you can see here, chromatography is a very important tool to scientists. But
that's all the time we have for today. I'm Jason Steiner, and this has been Everyday Science.